pleasure to introduce Dr. Xionghao Lin. Dr. Lin received his PhD degree in pharmacognosy from Peking University in China. In 2013, he joined Dr. Simon Wang laboratory at Howard University as a postdoctoral fellow to work on novel selective H-TAC2 inhibitors targeting Alzheimer's disease. Later, he joined the RCMI proteomics facility directed by Dr. Xionghao, where Dr. Lin focused his research on developing selective transcription inhibitors for HIV and Ebola viruses. He oversees the mass spectrometer. We have a new one, Orbitrap Explorer mass spectrometer, and he has been developing qualitative and quantitative proteomics methods. Dr. Lin also provides proteomic services to researchers from other institutions. We focus on HIV-funded projects. Since 2019, Dr. Lin became a research assistant professor at Howard University College of Dentistry, where he teaches undergraduate courses. Same year, Dr. Lin received a DC CFAR Pilot Award and RCMI Investigator Development Core Grant to support his HIV research. He has published 38 peer-reviewed articles, and during last year, he became a topic editor for the journal Frontiers in Pharmacology. Today's seminar is titled Protein Phosphatase 1 Targeting Small Molecules for HIV Transcription Inhibition, and this project aims to find selective HIV-1 transcription inhibitors to target the latent virus reactivation. So basically, for future improvement of combination of antiretroviral therapy. So, Dr. Lin, please, welcome.
Good afternoon, everyone, and thank Dr. Masova for the kind introduction. And as she mentioned, today I'm going to talk about protein phosphatase 1, PP1, targeting small molecules for HIV transcription inhibition. Yeah, to start off, I would like to give some background. So there are over 1.1 people living with HIV in United States, which is about 0.35% of US population. And every year, there are over 36,000 people diagnosed as new HIV, and which is about 3% of HIV population. And over like 15,000 people died from HIV. Therefore, there's still increasing as a total HIV population is still increasing every year. So the most affected subpopulations are African American, male to male sexual contact, and followed by Hispanic and white male to male sexual contact. And according to the data, minorities in United States account for about 69% of annual new HIV diagnosis, with 31% of the population. And the ultimate goal for HIV treatment is to find a cure. So in September this year, FDA approved a first trial investigating CRISPR gene editing as a HIV cure. And in 2014, United Nations AIDS program set ambitious goals for ending the HIV epidemic. And the first goal is to reach 90 by 2020. So 90% of people living with HIV are knowing their HIV status, and 90% of people who know their status would receive the treatment, and 90% of people on treatment with suppressed viral loads. And also the goal try to reduce the annual number of new HIV infections among adults to 500,000 worldwide, and also to eliminate the discrimination. It turns out only 2 countries, Botswana and Australia, met the 2020 goals. So the key to success was providing the comprehensive HIV testing and treatment programs. So in United States, we almost reached like 85, but also depending on different cities, like Mississippi is much lower, and New York City also met the 90 goals. So which means most of countries, we haven't reached the goal 2020. It also means we have more work to do to reach the next goal, 95 by 2030. And then this goal would want to further reduce the annual number of new HIV infections among adults to 200,000 worldwide and eliminate the discrimination. And to achieve this goal, we need to provide or optimize a comprehensive HIV therapy, which includes the rapid HIV testing. That's the key. We have to know who are infected, and then we empower physicians to initiate the treatment as soon as possible. Also achieve and maintains the viral suppression, offering simple administration. So current HIV drug has to be taken every day. If we can develop the long-acting drugs, the drug to prevent the viral reactivation, outer interruption of treatment, and also develop antibody to reduce the drug administration frequency. That would be a great help. And also, we would like to support the management across different life stage, especially aging HIV population, because now people can live longer due to the success of antiretroviral therapy. And we also would like to accommodate a wide range of treatment considerations, design the drug to target different steps of HIV replication and to increase the barrier for drug resistance. And here is the HIV genome and the structure. So the latent HIV RNA genome includes 3 major genes, GAG, POL, and the envelope gene. So the GAG gene encodes the protective core capsid and also the matrix proteins for viral assembly and infection. And the POL gene encodes for the reverse transcriptase, integrase, and protease. Basically, they are essential enzymes for reverse transcription, genomic integration, and viral maturation. And then the envelope gene encodes the GP120, the surface glycoprotein GP120, and the GP41 that determine the viral entry into the cells. And the HIV genome also contains 2 important regulatory genes, RIF, TET, and RIF, that activate HIV transcription, as well as 4 accessory proteins, WIF, VPR, VPU, and the NEF. They are less essential for the viral replication, but they are essential for the viral pathogenesis. And here is the HIV life cycle. To establish infection, the HIV must first attach to the host cells. Attachment occurs by the interaction between GP120 by the surface of virus and the CD4 receptor on the surface of host cells. And besides CD4 receptor, there must also be a co-receptor CCR5 and CXCR4. And once the outer attachment, the viral envelope will fuse to the cell membrane, and then the viral RNA will be released into cytoplasma, and reverse transcriptase makes a DNA copy out of RNA strand. And once the DNA strand is formed, the reverse transcriptase would degrade the RNA string, and a complementary DNA string will be added by reverse transcriptase, resulting in a double-stranded DNA, which would join the non-covalently to form the circular DNA, and then the circular DNA will be imported into nucleus and integrated into human genome by the integrase. And the integrate DNA is referred as a pro-viral DNA, which could remain dormant, or we also call it latent, or with host cell activation, the pro-viral DNA could be transcribed into a messenger RNA. And then viral messenger RNA will be further translated into viral enzymes and structure proteins. And then the variants will be released by budding. And some of functional proteins will be formed by cleavage of a lone polyprotein by protease during the vitral maturation. And currently, there are FDA drugs to block the viral entry and inhibit the reverse transcriptase, integrase, and protease. However, there's still no available drug to target HIV transcription, which is essential for the latent HIV reactivation. And that's why our group has been focusing on develop selective HIV transcription inhibitors. And targeting HIV transcription can be challenging because at this stage, the viral genome has been incorporated into human genome. And it's difficult to find the target or drug that specifically inhibit viral transcription rather than host transcription. And let's look at how the HIV transcription is activated. So based on our previous studies and also research from other groups, HIV TET protein interacts with PP1 through RVxF binding pocket. And they work together to recruit the positive elongation, transcription elongation factor B, including CDK9 and cyclin T1 from 7-SK as an RNA complex to HIV RNA to form a super elongation complex that phosphorylates negative elongation factor, deceive, and the C-terminal domain of RNA polymerase, thereby activating HIV transcription. And there is a huge difference in HIV transcription without HIV TET or with HIV TET. Without TET, the transcription can be initiated, but it only produces short abortive transcripts that do not support viral replication. And with TET, HIV transcription can be activated more than 100-fold. Of course, HIV activation is way more complicated than what I'm showing here, but we have proved that both PP1 and the TET play essential roles in HIV transactivation process. And here shows how PP1 and the TET interact with each other through the RVxF binding pocket, and both of them induce the HIV transcription. We can see that's an induction here. So based on this information, the idea we came up with is to design small molecules to bind to the RVxF pocket of PP1 so that we can disrupt PP1-TET interaction, thereby blocking HIV transactivation. And to do so, we screened the small molecules binding to the RVxF pocket of PP1, and we identified the initial compound 1703 for HIV inhibition, and we confirmed this molecule binding to the RVxF pocket of PP1. And however, we found there's a metabolic instability issue for this molecule. That's why we further optimize this molecule to receive a new analog, HU1A, which showed improved HIV inhibitory activity and also metabolic stability compared to 1703. And in the following slides, I would like to introduce each part more detailedly. So first, to identify PP1 binding molecules for HIV inhibition. So we use PP1 in the RVxF peptide from TED to build a computational model for the high throughput virtual screening of small molecules. And we screened 300,000 molecules from enamine chemical database. Then we received 262 candidate compounds, and they were further tested for HIV inhibition using a pseudotyped luciferase VFVG virus. And pseudotyped virus is worth noting that they only carry out one round HIV infection because the NEF gene has been replaced by luciferase reporter gene, and they also have a frame shift on the envelope gene. And as a result, we identified compound 1H4 that inhibit HIV replication with IC50 at 10 micromolar, CC50 at 100 micromolar. And then we synthesize analogs of 1H4 to perform the structure activity relationship analysis and to receive a better analog 1E703 that inhibit HIV replication at 5 micromolar and the CC50 over 100 micromolar. And we found compound 1E73 inhibits HIV replication by specifically targeting HIV transcription rather than reverse transcription and translation. And here is the HIV inhibitory and the toxicity data of 1E703. And then we see E703 inhibit HIV, TED-mediated HIV transcription, but we didn't see any effect on any inhibition on the reverse transcription and the translation. And then to test if compound 1E73 binds to PP1, we use surface plasma resonance SPR technique. So we immobilize the recombinant PP1 on the chip and then let the 1E73 containing buffer pass through the flow channel. If compound binds to PP1, would change the polarized light refraction angle, which could be detected by the instrument and generate the dynamic binding curves. And as shown the result here, E73 binds to PP1 with binding affinity at 6 micromolar. And we also shown that compound 1E73 can compete with PRB TET peptide, a non-peptide binding to PP1 RVxF site, compared to DMSO control, because there's no much effect on the PP1 TET binding for DMSO control. So now we have confirmed the compound 1E73 binds to PP1, but we don't know why it's the exact binding site. And to identify the exact binding pocket on PP1, we developed a new method called protein painting. The idea for protein painting is that PP1 itself can be digested by trypsin, and then we can detect the peptides from PP1 using a mass spec. And if we incubate PP1 with painting molecule, and that binds to lysine and arginine on the surface of PP1 covalently, which would block the trypsin digestion, we would not be able to detect the peptides from PP1. And if we incubate PP1 with compound 1E73 first, the compound binding would compete painting molecule out of binding pocket, because compound binding is a non-covalent binding. Outer dissociation would expose the binding pocket to trypsin digestion, so that we can only detect the peptides from binding area of PP1. And here is the result. We can see the E73 treatment can rescue a few peptides from protein painting, and especially the red peptides, the yellow peptides here. And here is the top peptides protected by compound 1E73 treatment. We can see the signal were rescued more than 70%. And we map the trypsin digestion size on the surface of PP1 in blue color. And we also map the compound protected size on the surface of PP1 in red color. And first, we can exclude the arginine 221 because it's located nearby the catalytic site of PP1, and we have proof that compound E73 has no effect on PP1 catalytic activity. And other protected sites are located on the side and on the back side of catalytic site of PP1. And then to better understand the possible binding between PP1 and E73, we performed the molecular docking, and 295 configurations were generated and classified into 13 clusters based on their binding energy. And then together with pending data, we received 3 potential binding sites, including MYPT, which is a protein binding companion of PP1, and we found lysine 234 and 238 around this binding pocket. And then we also found the lysine 168 around the RVxF binding pocket, and then also arginine 187 and 188 around the NIP1 helix PP1 interface. Since based on our previous FAR study on Y701H4, we found out the ketone motif is important for its HIV inhibitory activity, but we did not observe any interaction between E703 ketone motif and the PP1 in the MYPT pocket, suggesting that MYPT pocket is less likely to be the primary binding target of Y703. And we did observe the hydrogen bond interaction between ketone motif of Y703 and PP1, both in RVxF pocket and also the NIP1 PP1 interface pocket. Therefore, we consider those 2 pockets as primary PP1 binding pocket of Y703. And for further validation, we use PP1 and the central domain of NIP1 as a model. They interact each other through 5' RVxF and the NIP1 helix motifs. So we fused PP1 to the we call this system as a nanobit split protein-protein interaction system. We fused the PP1 to the large bit of luciferase, and then we fused the CD-NIP and its mutants, different mutants on RVxF and the NIP1 helix to small bit of luciferase. And if they interact with each other, we'll bring the large and small bit together to regenerate the luminescence signal. And then we treat the compound once E73 on different complex. The compound would be able to disrupt the interaction between PP1 and CD-NIP or their mutants. And as a result shown here, so the RVxF mutant eliminate the disruption of compound Y73 on PP1 CD-NIP interaction compared to CD-NIP mutant. And we also see the CD-NIP helix mutant has a slight effect on the interaction of PP1 and CD-NIP disrupted by compound treatment. So therefore, we consider E703 binds to both RVxF and the NIP1 helix pockets of PP1 with RVxF as a primary binding pocket of Y703. And next, we test our Y703 compound for HIV inhibition in humanized HIV-infected humanized mice. So the compound can reduce the HIV tRNA by 40-fold. And then we also perform the drug metabolism and the pharmacokinetic study for this compound. And what we found out is this compound is not stable in the mouse plasma. It degrades into many degradation products. And then DP1, the PK data show DP1 and the DP3 are the major metabolites of E703. And then from PK study, we can see E703 converted into DP1 in DP3 less than 30 minutes. And we also synthesized these two major metabolites and tested them for the HIV inhibitory activity. It turns out both of them are much less active than the original compound 1703. And to figure out why they are much less active, first we check their PP1 binding. And it turns out both compounds can still bind to PP1 pretty well, even a little bit better than original compound 1703. And both of them also can compete with PRB-TET peptide on PP1 binding, which suggests that PP1 binding is not the reason to cause their loss on HIV inhibitory activity. And eventually we found out that they lost their HIV inhibitory activity because they have a poor cell permeability. And we can see E703 can get into the cells pretty well. And then DP1 only have a small portion to get into the cells. And then DP3 was not cell permeable at all. That's the reason they showed much less active than original compound 1703. And since we have identified the ester bond and the amine bond in the ketone motif in E703, the bio size of 1703, and based on our previous SAR study, we also found R1 and R2 group modification are also essential for the HIV inhibitory activity of 1703. And so we synthesize the 49 analogs by replacing the linker and also the R1 and the R2 groups. And we actually synthesize more than 49. We confirm the 49 analogs with over 95% purity, and we also confirm that structure using the NMR and the mass spec. And then we test those 49 analogs for the HIV inhibition using a pseudotype HIV luciferase VFBG virus for HIV inhibition, and also monitored the cytotoxicity. And also the selective index between activity and toxicity also calculated. And there are few compounds shows pretty good selective inhibitory index on HIV. And here has some few analogs. We received a good inhibitory index, but they only show the moderate HIV inhibition. So the good selective index were contributed by their promotion on the cell proliferation. So that's why we chose the compound here as the top candidates for further study. So we perform a dose-dependent analysis for those top candidates. It turns out the compound HU1A is the best candidate in terms of its HIV inhibitory activity and also the cytotoxicity. And we compare this compound side by side with 1703. We can see we replace the ester bond with the amine bond, and we also replace the ketone motif with the urea motif, and we also modify the tail R2 group of 1703. And to further confirm the HIV inhibitory activity of HU1A, we use a competent HIV-3B virus. We call it competent because it's different from the pseudovirus. This virus is competent. It can continually infect the new cells. And as shown here, the new analogs shows better inhibition on the HIV GAG mRNA and the P24 expression compared to 1703. And the new compound also showed improved metabolic stability compared to 1703, especially in mouse plasma. We can see compound 1703 degraded in 30 minutes, and then this compound is totally stable in the mouse plasma. And we also see there's a huge difference in the human liver microsome. We can see the new analog shows much better stability compared to 1E73. And last, we would like to see if the new analog still bind to the PP1 RVxF pocket. And then we use the same nanosplit system, and we treat compound new analog, and we can see that the RVxF mutation mainly eliminate the PP1 CD-NIP interaction disrupted by compound treatment. And we also dock this molecule into the RVxF pocket of PP1, and we can see the urea motif still remain the interaction with the same residues, leucine and asparic acid of PP1, similar as 1E73. And the mechanism for both molecules for HIV transcription inhibition could be both molecules bind to the RVxF pocket of PP1. That would disrupt the PP1 TET interaction and then block the whole HIV transcription process. And taken together, so through our project, we have proved that PP1 RVxF site can serve as a potential drug target for HIV transcription inhibition. And we developed the compounds 1E73 and new analog HU1A as HIV transcription inhibitors. And we also developed several new techniques, including protein painting and nanobit for protein ligand binding or protein-protein interaction. So in future, we would like to figure out how host factors involved in PP1 pathway affected by 1E73, because HIV also hijack a lot of host factors for its replication during HIV transcription. And we also would like to see how those new compounds affect latent HIV viruses. And eventually we would like to apply those compounds along with the current antiviral drugs. And last, I would like to thank my lab members, especially my mentor, Dr. Nikai, who had a great contribution on experiment design and method development. And Mr. Wang helped with the HIV activity screening, and also Dr. Ivanov helped build up the nano split system, and Dr. Ahmed helped do the nanobit split assay, and Dr. Kumari helped with the GAG mRNA and the P24 measurement, and Dr. Amasova helped with the cell permeability study. And I would also like to thank the funding support from NIH to Sergei Nikai, to Dr. Nikai, and also a DCC4 pilot project to myself. And thank you all for your attention. I'm happy to take any questions.
Nice talk. I have a question. So you've done some toxicity on the compounds, but have you done long-term toxicity?
So the cell treatment we only did like about a week, but we did inject the compounds to the mice, like when we were doing the HIV activity testing. It didn't show, we didn't observe the toxicity. Like we did a 1 week injection, like every other day we did the injection.
No, I was more curious about the toxicity in vitro, the long term, because your compounds, I'm sure that for the effect on latent, you are trying to go into the block and lock strategy, similar to the DCA, the one that Susana Valente has been working. Do you think that is a similar type of molecule?
It's different. I did see some kind of like combination treatment with the current drug, and we also compare. We didn't do long-term treatment, but I did compare the toxicity of the E73 compound with the current anti-HIV drug, and it turns out that some of like reverse transcriptase actually pretty toxic compared to our compound. Integrase, we did see integrase. That's really nice on toxicity. Basically, I didn't observe the toxicity from integrase.
No, well, I mean, I don't know if you're familiar, but if not, we can talk even offline of the studies by Susana Valente, where they have a TAT inhibitor, where they culture lethal infected cells with this inhibitor that is called DCA. I don't know the spell. And after a while in vitro, and what it does is it changes the promoter of HIV and it represses transcription. And it's kind of the block and log strategy. And I don't know if you plan to do those studies with this compound.
Yeah, I read that paper. They did about kind of like a year treatment, basically. Like they start the antiviral, kind of like they did treatment using the antiviral drug together with the DCA, and then after several months they stopped treating. They also like select the mutants, HIV mutant, to check like how the virus react to the drug treatment.
But in vitro, I think they use some cell lines like GLAD, where they culture the latent infected cells. So then what they show is that after a while, those cells, because you are inhibiting TAT,
yes,
it makes the cells more refractory to reactivation, right? And I wonder if your compound is also going to do that and to be kind of another candidate for the block and lock strategy that.
I haven't figured out like how to do a long term, like about a year, because if we use the live virus, they are going to keep infecting the cells, right?
But you can choose, so there are these latent infected cell lines. I mean, as a proof of principle, right? They did it first on cell lines that are latent infected with HIV, and you can stimulate the virus. But after culturing these cells just with the inhibitor of TAT inhibitor, the cells will become refractory to reactivation, and because it changes the how the chromatin is compacted around the HIV promoter. And I wonder if, like, that is the proof of principle, and then they've done multiple other assays, studies, including monkey studies and everything. But I wonder if your molecule will also be kind of doing a similar effect, that after a long time in culture, the cells will become more refractory to reactivation.
I see.
I don't know. I mean, you thought about that, because it looks like a nice candidate for the block and lock strategy, right?
Yeah, thank you.
Cure.
Yeah, thank you for a suggestion.
Are there any other questions? Okay, hearing none, I think we can conclude this session. I want to thank Dr. Lin and Dr. Amasova again. And this seminar recording will be available on the CFAR website and YouTube channel in a couple of days. And thank you all so much for attending.
Thank you.
Yeah, thank you.
Thank you.